Hi there guys, and welcome to another A-level biology video. Mr Phillips, and today we're looking at the nervous system area and behaviour and responses module, and we're looking at 6A.1 stimulus and responses, and particularly looking at the reflex arc today. So first of all, before we look at the reflex arc, we just need to understand, why is it so vital to survive? Well, hopefully this is relatively obvious for you. Why does responding allow survival? Well, one, it allows you to find food. So if you find the food, you can respond to it. One, it avoids predation. One, it avoids harmful chemicals. It can help you keep warm by detecting the cold and responding to it. And basically, it helps us find favourable conditions. And this is a good method of survival for any species, because if you can't do this, you will die out. And effectively, there's 3 steps to it. There's detection of the stimulus, which might be any type of energy change. And then there's the coordination, which involves your nerves and the brain. Not always the brain, obviously, because it might be an organism that doesn't have a brain. And then there is the action, which is normally an effector causing a response. And the effectors are normally a hormone or a gland which releases hormones, or a muscle. So we're going to look at this next time in a bigger video, really, but just introduce what receptors are. Receptors are specialized cells, and they cause the action potential. Okay, so there's the cells that basically receive and detect the change in the stimulus, and they cause the action potential to be fired, so they cause the response to happen. They're found in the sense organs, and they, as I said, detect normally one specific stimuli. So, for example, the receptors in your retina detect the changes in the light that enters your eye, and you have baroreceptors in your arteries and veins, which detect the change in blood pressure. So some are specialised cells, and they'll generate an action potential. Others simply, it's the end of a sensory neuron, and that can trigger an action potential. But the word action potential is one we'll look at in a lot more detail in a future video. So effectively, again, overview of this is you get stimulus, which is a change in the internal and external environment, often an energy change. The receptors are specialized cells that detect the stimulus, and then it is the effectors that cause the action, the follow-up, which is the muscle and gland. But we need to know what is happening in the middle and what's happening with the quarters. So if we have a look at this, and again, just different way of showing it you. So your nervous system effectively causes a feedback loop, often negative feedback. So you get an input, which is detected by a receptor, so the temperature's gone up. Your brain coordinates a response, and that causes the effectors to respond, and that lowers the temperature, and then that's detected again, and then it keeps responding. So it's something that's called negative feedback, which is detecting a change or reversing a change in the stimulus. And basically it does these three things all the time, and that causes us to have this sort of constant level in our body. So this picture shows you from an A-level book what is the typical simple reflex, or the reflex arc is in humans, but we're going to have a look at it in a couple of different ways. So let's just sort of go through it and just see whether you're happy with this. And really, this is GCSE stuff, so you should hopefully be relatively happy. So your stimulus here is heat, so that's a change in energy. The increase in energy of heat goes up. That is then detected by pain receptors, so that's your receptor cells in the skin, and the skin is an organ, a sense organ. Your sensory neuron then generates an action potential, or nerve impulse as we'll call it at the moment, and that goes up the sensory neuron, up your arm. That then gets to the spinal cord, and in the spinal cord, or the central nervous system as this area is, you have another neuron called a relay neuron. And your relay neuron, to get from the sensory neuron to the relay neuron, you have got to jump across a synapse, which we'll look at in a couple of lessons time. And then to get from the relay neuron into the motor neuron, you've got to go across another synapse going to chemical messaging, and then it goes down the motor neuron. Now the motor neuron is then going to connect, and it's going to connect to the effectors. Okay, and the effectors are, in this case, it's going to be a muscle in your arm, which will cause you to retract your hand from the hot thing you're touching. but effectively it could also be a gland as well to release hormones. Let's say if it was, you might release some adrenaline from your adrenal glands at the same time. Now this doesn't have to involve the brain, but you do normally send a message to the brain as well, and that means you can prevent it from happening. And the reason it doesn't involve the brain is it gives you a few less seconds to respond, and therefore it's a quicker response, and therefore you're less likely to get damaged. So from GCSE you probably learned something like this, and again you must learn the stages here. So you get the receptor, which is detecting the change in stimulus. That goes to the sensory neuron, which is often connected directly to. That then goes to the relay neuron via a synapse. Synapse then often connects the relay neuron to the motor neuron. That affects the effector, which in this case is the muscle, and then that drops the plate in this example. And the central nervous system is involved, and that is the brain and the spinal cord. Messages can be sent to the brain, but we don't need the brain in order to respond, but the brain is able to override if needed. And you might get an example like this. So again, exactly the same thing, just a different sort of version of it. So you have the receptor in the skin, sensory neuron, relay neuron, motor neuron, and effector. Now there are lots of different types of behaviour and responses that we can have. So you could have a question on this in an exam, and often a lot of psychology sort of these areas. So real simple behaviours are kinesis, and these are non-directional responses to a stimuli. So for example, you've got here the rate of movement of a woodlice. It's not directional. If you put woodlice in unfavourable conditions, as this dry conditioned petri dish shows here, they will continue to move fastly, and they're only moving fastly as soon as they find favourable conditions. They prefer moist and dark conditions. They will slow down and stop moving. So they're not actually searching actively for those moist conditions. What they're doing is just moving fast until they find those moist conditions by chance. Another example of this is the planarian or the flatworm, which will lie still on the inside of rocks because they're favourable conditions in the dark and damp conditions. But if you turn the rock over so they're in the sun, they will continue to move randomly as fast as they can until they randomly hit dark shaded conditions again. And basically this is a very simple behaviour type that prevents them from just sitting and stopping in exposed areas. They'll continue to move until they're in safer, more favourable conditions. And then a bit more advanced is something called taxis. I always think of this as you get a taxi to go somewhere particularly. So this is a directional response to a stimulus. So for example, something like the euglena, which is a type of algae, will swim towards light which is called positive phototaxis, and it will do this because the euglena has chloroplast, so it wants to do a photosynthesis, so therefore it does positive phototaxis, swimming towards the light to gain the light that it needs and aid its survival. And a lot of organisms will do this with chemicals, so we'll move towards good chemicals, positive phototaxi, positive chemotaxis, and it'll move away from chemicals that are harmful, and that is pretty logical and obviously a good survival mechanism. Now you can investigate this through an investigation called a choice chamber. And a choice chamber effectively is where you can set up different environmental conditions to investigate the behaviour of either taxis or kinesis of different organisms. So for here you can see in this example we've got some results. We've got the woodlice, and the woodlice are going to be put in the hole at the top of the chamber here, and then effectively then what they're going to see is that the woodlice will go either into the bright light or into the shaded light, and we look at that after 5 minutes in this investigation. So what you can see is you would expect more woodlice to end up in the shaded side overall. As you can see, does happen here, doesn't happen the second one, does happen, does happen, does happen, equal that one, doesn't happen there, does happen, does happen, and does happen there. And the reason you expect that is because they move fast whilst they're in the the light because it's unfavourable conditions, and they'll slow down when they're in the least favourable conditions. But because it is a random effect and they're just moving randomly, they will still obviously venture out into the light, and then they'll speed up again until they move back into the darker conditions. So in terms of setting up this experiment, how would you do it? Well, what you would do is you would put half of the Petri dish with dark paper, and half the Petri dish would obviously have no paper over the top of it. And what you could do is put some damp filter paper in both sides to control the humidity. So if you're just looking at light intensity, you'd put some damp light paper or cloth on the bottom to make sure the humidity is the same. And then what you'd do is you'd place 10 wood lice maybe in the centre of the mesh of the chamber, and in the centre, and you'd put it up, and then you'd close the lid and leave the wood lice to go. So after 5 minutes, then you'd take off the lid, record the number of wood lice on each side of the chamber, and you'd try to minimise the time the lid is off so the environmental conditions would stay the same. Then obviously you'd repeat the experiment, gently moving the wood lice to the centre again, and you could use a small soft paintbrush maybe to do that. And effectively that's what you would do. So you'd be looking at the movement of the wood lice. So you can see these trials, this is where the wood lice end up. There isn't like they learn. You can see here that the wood lice don't get any better. The wood lice just simply keep moving randomly, and they go faster when it's in non-favourable conditions, i.e. when it's in light, and they get less fast when it's in darker conditions.